Hi, everyone, and welcome. We're really glad you could join us today. My name is Beth White, and I'm the Director of Graduate Enrollment in the medical school, which means that I work broadly with our master's and PhD programs and issues related to their students. But today, we're here to talk about one of those specific programs, the Structural Biology Master's Program. So I am going to turn this over to Dr. Andy Gulick, who is the Program Director.
Great. Thanks, Beth. And hello, everyone. Hope you're having a good day on this snowy day. What I'd like to do over the next few minutes is sort of tell you a little bit about our program in structural biology, the master's program. So you can probably find information on the website, but my contact information is here and our website is here. So I will encourage you to go there, and you can find again, how to reach me if you have questions or other information if there's other things that you're interested in. So the plan is to basically tell you about our program, and what I'm going to do is sort of do this in two steps. First, just sort of talk about structural biology as a whole, and then we'll talk a little bit about the actual program and what we're expecting out of you and what you can expect out of this program. And then I didn't mention it here, but I have a few slides at the end about applying to the program and sort of the things that we'll be looking for. Okay, so my first question is what is structural biology? And this is something that has gotten a lot of prominence over the last few years, unfortunately, I guess. But we've all become very familiar at looking at protein structures and molecular structures. We see them all the time on the news and in the media. And so this is really something that in the past we kind of went into more detail about showing people what we're looking at, but now we're all really comfortable with seeing images like this, which are actual molecular structures of important biological macromolecules. And so, we think about structural biology as an interdisciplinary science that sort of crosses the boundaries of biochemistry and pharmacology. We think about chemistry and chemical biology. And then for more sort of mathematically inclined people, the world of structural biology involves biophysics and math and computer sciences, and even some other sort of things about image analysis and sort of like big data. So it's really a way of sort of thinking about biological problems and biological processes at the molecular level, right? So perhaps maybe one way to think about this is what structural biology is not, right? And so you've probably, if you're here, you've taken some biology classes and you've seen schematic diagrams that represent important processes. So this is showing the release of neurotransmitters at the synapse, and neurotransmitters coming out of one cell and then binding to receptor molecules on another cell. And this is obviously very important. There are people that sort of can spend their career sort of thinking about these processes at this level. And the world of structural biology is really to take it a step further and dig a little bit more deeply into it and really sort of understand the molecular interactions that govern biological processes like this. So how do neurotransmitters bind? How do they generate the ion transport across the membrane? Are there ways that we can manipulate the system or develop molecules that could block the process? And this is really where the world of structural biology gets into this. So thinking about things not even at the cellular level, but down at the molecular level. And again, this is where all of the tools from the previous slide really come into play. So using biochemistry and chemistry and biophysics, and all these different techniques that can allow us to to then visualize these processes at the molecular level. Okay, so let me slide this over to the side a little bit. So what is structural biology? It's a desire to understand important processes at the molecular level. We do a lot of actually visualizing proteins and other macromolecules at the atomic level. So there's a number of different techniques that we can use. And we do this so that we can actually determine the atomic structure of these important molecules. We can additionally, if we sort of understand these processes well enough, we can begin to predict what proteins will look like using just the DNA sequences. You're again probably aware that there are many more DNA sequences that encode proteins than we know of the actual protein structures. And so by looking at the many DNA sequences, we can infer the necessary protein sequence. And in many cases, we can then use what we know about related proteins or some additional computational techniques to predict what the protein looks like. And so I've talked a lot about sort of just generating these proteins, but or generating these structures of these proteins, but it's important to realize that this is sort of just the first step. And so really what we want to do is use these structures to then inform our understanding of the biological processes. So we could, for example, use those insights to, as I said, predict the function of uncharacterized proteins, or we could use it to engineer proteins to have novel activities or to adopt different structures. Or again, we can use our knowledge of the structures of proteins to understand their dynamics, their interactions with other proteins. Again, whether that's within the same organism or if it's a pathogen interacting with host proteins, for example. Again, using the SARS virus as a great example. Or we could understand evolutionary relationships as well. So we know that there's often a common underlying sequence that is shared among different closely or sometimes distantly related organisms. And so understanding the structural features that were maintained among different organisms and sort of how that can inform our understanding of the evolutionary relationships between organisms and sort of how they've adapted to their different environments. What we will do within our program, as we'll see when we go over the curriculum, there's both courses that you will take that will build upon your studies that you've done as an undergrad, and then laboratory research. And so our program will really prepare you to develop skills in a variety of techniques, again, both structural, sort of determining structures, and then analytical skills to study important biological macromolecules, and again, mostly proteins, but there's other macromolecules as well that fit into our world of study. A lot of these skills are things that are highly in demand in both biotech and the world of pharmaceutical companies. Even just being able to produce proteins at sort of very high levels and high purity and high activity is a really sought after skill. So there's I think a lot of people develop sort of the molecular biology skills, and these are things that you may do in some preliminary undergraduate labs. But a lot of the protein work actually takes sort of just longer to do and maybe isn't quite as suitable for a lab. And so, belonging to a program where you're going to develop those skills is sort of really valuable and perhaps a little more unique. These, the time spent in the master's program could strengthen your application to a PhD program if that's what you're interested in. And then, again, we'll build your research resume and give you exposure to a lot of different types of techniques. Okay, and so as I said, we've through this program, you will build on a lot of the biochemistry that you may have had as an undergrad, and will get exposure to a lot of other techniques: protein purification and production, and then protein structure determination and analysis. Okay, and so our program is focused on hypothesis-driven research. So the different faculty in our department all have different sort of areas of study that they're interested in, but largely, we use a sort of shared core set of techniques that allow us to determine structures and investigate the structure-function relationships of important biomedical proteins. And then, again, after determining the structures, we then move on to hypothesis testing experiments. So we may understand the role of a protein or different parts of a protein and see if we can make changes into them to study how they work. We can try and inhibit our proteins using small molecules or drug compounds to understand how to build those molecules and maybe even how to make them work better. And then, as I said, we can understand how changing the protein sequence and protein structure influence the function of the protein, of the cell that contains that, and in some cases even of the entire organism. Okay, so those are a lot of sort of broader sort of sense of what you'll be doing. And so here I actually put like some real explicit skills of types of experiments that students that go through our department can develop expertise at. And so these include sort of molecular biology tools to generate DNA constructs that we can use to drive protein production. A variety of different protein production systems, including bacterial and eukaryotic and different types of cell lines. And then, as I said, for people in our department, a lot of what we do is to analyze protein structures and protein activity. And so producing highly pure and large quantities of protein is something we do day to day. So people in our labs tend to become real experts at making protein. We also do a lot of biochemical assays, and so analyzing proteins for how they function. One of the main ways that protein structures are determined is to grow crystals of proteins. I had some crystals on one of the earlier slides. And so students in our department will have the opportunity to crystallize proteins, and in some cases actually solve the structures through a technique known as X-ray crystallography, in which the crystals are exposed to X-rays, and the X-rays scatter off those crystals in a way that is dependent on the underlying atomic structure. So that sort of is where these structures come from. We do, as I said, a lot of computational studies as well to look at related protein sequences and protein structures in order to understand how those differences relate to differences at the functional level. We do, work with either medicinal chemists or other pharmacologists to understand how small molecules can bind to the protein and in some cases be blocking protein activity, and so we can identify or develop new drugs or drug-like molecules. And then, people in our lab, we've got sort of different types of labs, some that are, really focused on important biological problems and using existing structural techniques to understand those problems. And then we also have people in our department who are developing new tools and new experimental and computational techniques to analyze other protein structures. So they may not have a particular system or protein that they're focused on, and really what they're doing is developing the tools. And the presence of both of those types of scientists in our one department, I think, is another strength of what we do. So again, then, some of the other explicit tools like molecular biology to generate mutants of proteins. And then, again, understanding protein structure and the relationship between structure and function to investigate uncharacterized proteins, and so on. Okay, so I hope that gives you a sense of what structural biology is about, and sort of the types of things that people that go through our department will learn, and the skills that you'll leave with. So I guess I'm going to switch gears here now to talk a little bit about our curriculum. And as I mentioned, it's a 2-year program, and there's really a total of 30 credits, and really the 2 fall semesters and the 2 spring semesters of your first and second year will look pretty similar, with the one exception of the courses that I'm highlighting in blue. So the generally speaking, the first year you may take sort of a introductory graduate biochemistry course. And in the second year, you'll take a crystallographic methods course that'll define for you some of the strategies for solving crystal structures. Again, depending on the project and the lab of sort of where somebody is coming in, we may switch which year you take those, again, depending on which is sort of more important to kind of get you prepared to carry out your thesis research. And then in the spring semester, we have 2 courses, one in principles of protein production and analysis that talks about all different ways to make proteins and purify them, and then analyze them, again, both structurally and functionally. And then a course that runs, again, in the spring semester on structural pharmacology that looks closely at important proteins that bind to important drugs, and understand sort of how they work. And again, those 2 courses are sort of both set up, and depending on your interests and the lab that you rotate into or place yourself into, would sort of dictate which of those you take in which semester. So then all 4 semesters, you will take a structural biology seminar course, which is a 1-credit course in which students each present twice. So they present 2 papers from the literature in sort of the early stages. And then, maybe by your second year, the students would be presenting 1 paper and then an update on their research that they've been doing in the lab. And then we also have research credits that you will take to perform your thesis research. And again, this will be carried out every semester, so all 4 semesters in the program. So the last course that I'll mention is this 1 credit course that you take in the spring semester of your first year. And this is essentially a thesis proposal course in which you will prepare for your master's committee a document that sort of describes a research plan. And it's largely going to cover, the preliminary work that you've been doing up to that time, and then your plan moving forward. So it is something that allows for you to show that you sort of thought independently about your work, and you can sort of capture a large project and put it together in a way that makes sense for other members of the department. Okay, so all told, that adds up to 30 credits. It's 7 or 8 credits each of the 4 semesters.
Can I ask a question, Dr. Gulick, just based
Sure.
on what I've heard students ask in previous webinars? So this program is thesis only, so students don't have the option of capstone or thesis. It's a full-on 2-year thesis program?
Correct. Yeah. We've sort of thought about creating that kind of an option, and we haven't quite done it yet. And so we may, and we understand the appeal to that, and it's but as it currently stands, this is what it is. It's the thesis.
And does that mean, do you think that most of your graduates would then go on to PhD research, or are there does this prepare you also for industry positions or some things beyond the academic track?
Yeah, so I strongly believe it positions you nicely for both. I mean, so as I was saying earlier, some of these wet lab skills are things that are highly sought after in industry, and really are not something that you get a whole lot of exposure to. There's techniques that I think people tend to do in classes, some of them molecular biology, PCR, running DNA gels. So those are things that, as I said, just sort of fit nicely in a project for a lab, in a lab class, or even something sort of smaller. If you did say an undergraduate research project, but things like purifying proteins and analyzing proteins, those are not quite as common, and I think that those are really good skills. So having those for industry, I think is very appealing. And then certainly if your interest was to then enter into a PhD program in, again, whether it's in biochemistry, in structural biology, in chemical biology, those are all tools that would be, put you in a really good position to rapidly succeed in a graduate program. Okay, good. Okay, so your thesis here is as we were just saying, is a major focus of the program. So you will complete a project with one of the within a lab, within one of the structural biology faculty members, and so what we like to do. So getting into some of the details about our department, we're kind of a small department with only a handful of faculty members. And so first of all, that means we're going to be, at least for the next few years, a relatively small master's program with 3 to maybe 5 students being admitted. And so, one of the things that you may want to do, and in fact, I'd encourage you to do, is to, again, look at our website, look at the faculty who are here, and think about if the work that they're doing interests you, and if that's something that you could see doing a research project with. And so if somebody is accepted into our program, they may identify, hey, I found this work really interesting, and I'd like to, learn more about it. And maybe after a conversation or two, you can sort of be admitted directly into that faculty member's lab as a master's student. Or we have also in the past done sort of laboratory rotations where somebody will spend time in a couple different labs and sort of get a sense of the flavor of the types of work that they do. And again, that will give you exposure to the types of research going on in the department. And work can be completely wet lab, laboratory-based experiments, or as I mentioned, we also have sort of more computational people. And point of fact is everybody does some degree of computational work, and so there may be computational opportunities even in the more traditional wet lab groups. Okay, so the last little bit is just to discuss your application, and sort of how we look at this. So these become kind of the three main criteria for how the department is going to review applications. We will look at your transcript, and again, we don't have a hard and fast GPA at which we won't look at you at all if it's below this, but we'll look at the overall. We'll look at your science and math grades, a little bit more focus, and then as I'm sure you've heard throughout your undergraduate career, we'll focus also on the trajectory, right? So we understand that the transition to college may not go as smoothly as perhaps everyone would like, and what we're looking for is that you sort of found your footing, and things are looking good for the last your junior and senior years. So don't be discouraged, but again, it is a factor that goes into convincing us that you're going to be successful in the master's program. We will solicit letters of recommendation, and again, looking for academic advisors or academic professors who have had you in their classes, mentors. Again, if you've done a research project as an undergrad, somebody that sort of worked with you closely and can attest to your work ethic and ability to analyze and think critically about scientific topics. And so again, if any of you are not seniors but are still at sort of earlier stages in your undergraduate career, develop those relationships with your instructors. Go to office hours and get to know people, and that's a good way to sort of prepare for the future when you're going to need letters of support from them. And then finally, we're sort of interested in knowing why you'd like to apply to our program, and what you think this will do for you. Tell us about your future plans. We're not going to hold you to them. We understand that plans always change, but we'd like to understand what your goals are for being part of the master's program, and sort of where you see this taking you. The statement is also a good place to sort of address, as I said, if you had sort of a slow start to your undergrad, or for whatever reason, maybe the grades aren't quite where you want them to be. We understand, and so use this as your opportunity to explain that. I think this is a good place for that. So, and then the last thing I wanted to mention here, and this relates to something I was saying a little bit earlier. So we are a small program. There's only a handful of us. You'll see if you go to the website and sort of look up our faculty. So feel free to reach out to one of us. Set up a whatever half-hour conversation to hear more about the type of work that we do, and the types of projects that we may have available for master's students. And if you do that, include that in your statement. I talked with Dr. Gulick and found his work to be interesting. And that will give us, again, a sense of sort of your interest level and your motivation. And it'll help too with that aspect of, do we do rotations, or are you sort of going to just directly enter right into a lab and start working on a research project? Okay, so the last little bit I had was some of the nuts and bolts of the application. So, you can find this on the UB website to apply. The submission deadline is April 1st. And as I said, line up your letter writers so that you'll get those letters of reference in place. We'll start looking at applications in April, and really start, getting back to people right then. So I think that was all that I had. Was there anything else? That was it. So I would be happy to answer any other questions about, again, either the field of structural biology, our program specific, or some of the administrative aspects.
Sure. I know I have a question that I know students typically have, and I'm wondering about in regards to those admissions criteria. How important is the level of research experience the student has had? And also, are GRE scores a factor? I know that students often wonder about that as well.
Yeah. So we don't require the GRE. If you have it and that supports your academic credentials, and you feel good about it, then by all means go ahead and submit them. If that balances, like I said, maybe your GPA in some way that you feel is important, go ahead. In terms of research experience, we view this as a research experience. So yes, while we'd love to know that you've done all these things before, it's not a requirement. You're coming here to learn how to do those things and to get that research experience to better position you to apply for a job in industry, or to apply for a PhD program. So if you already knew how to do all these things, then you'd be ready to go to them as it is now. So obviously, if you have experience, tell us about it in your statement. But if you don't, tell us about your favorite organic chemistry lab that you've done, or your favorite class that you took on evolutionary biology, and why that inspired you to think about DNA sequences and protein sequences of related proteins and so on.
Yeah, I think that's an important point. It goes back to sort of the holistic nature of our admissions process. I think in a lot of our departments, and it sounds like in yours as well. I think students sometimes are afraid because they don't check all those boxes and think they won't be a good applicant. But really, I think master's level study is intended to prepare students for whatever those next steps are. So
Yeah,
I'd encourage
absolutely.
students watching not to be intimidated too much by that process.
Yeah, I agree completely. So
great. Well, this has been really, really insightful. I've been interested to learn more about sort of what structural biology is all about. So this was really helpful. And hopefully those of you watching also learned a lot today from Dr. Glick's presentation. We encourage you to get ready to apply. We are still accepting applications for fall of 2022. So get those applications in, and hopefully we'll be hearing from you.
great.
Great.
Nice meeting you all.
Yeah, thanks so much for doing this, Dr. Gulick. We'll talk to you soon.
Sounds good.